Hey everybody, it's Standing for Truth. The debate that you are about to watch is between Dr. Charles Jackson and Dr. Herman Mays. I would say this is a must-watch debate. It's very engaging. A lot of great points are brought up, and these two gentlemen are both great debaters and both very knowledgeable. Unfortunately, this debate has only been available through Facebook, from my understanding. Therefore, Matt and I have put this together for those who may not have seen this debate or heard of this debate for YouTube. Guys, I hope you enjoy. And don't forget, please subscribe.
Just want to welcome everybody who's attending live, and we appreciate the support, and everybody who's watching via live stream, we thank you. And while there's a lot of familiar faces here this evening, I want to introduce myself. I'm Chan Arnett. I'm founder of Faithful Apologetics, which is a ministry that helps to equip Christians to fulfill the instruction given to us in 1 Peter 3:15, where he says, always be ready to make a defense to anyone who asks you for a reason of the hope that's within you, and to do it with gentleness and respect. So I want to start off with some thank yous. Obviously, thank everybody for attending. Also want to thank everybody who helped out, whether it was financially, whether it was with labor, or whether it was with prayer. We want to thank everybody who helped. And third, I want to thank the Marshall University Secular Student Alliance, who are here this evening and attended, and want to thank you guys, especially Rod Fuller. He was very instrumental in helping this go on. I appreciate his help. And this event was actually originally supposed to take place at Marshall University. But, what was it, a week and a half, 2 weeks before, Marshall sends us an email saying, oh yeah, we're going to be closed that evening. So we didn't have a venue. So the last thank you I want to give is to everybody at England Hill, especially Justin May and Nathan here for, they've put in a lot of work. So I want to give everybody a round of applause for them and for everybody that's helped. So I want to kick right into this, but I do want to say a few things. One is about debates in general. Now, something that's sorely missing in our culture these days is a searching for, a finding, and then an obeying of the truth. It's unfortunately, it seems like it's a lost art these days, unfortunately. And many of us suffer from what we call brand loyalty. You know, brand loyalty, where you just buy a product, whether it's the best product or not, you're loyal to that product. And you stick with it no matter what, right? There may be a product that's better, but you don't care, you want this brand, right? Well, unfortunately, what happens is many of us are brand loyal when it comes to what we believe. And I think that's unfortunate. We blindly roll with what we hear and what we see, and we never test our beliefs for their truth. Now, my hope tonight is that all of us, whether we're Christians or we're non-Christians, no matter what our worldview, is that we'll give up our brand loyalty for this evening and going forward, and we'll take in what each speaker has to say, take it seriously, process it, digest it, and test it for truth. And this is how events like this, these debates, become important and fruitful and very rewarding. Now, the title of tonight's debate is, does the genetic evidence support common design or common descent? Now, what do we mean by that? Well, retired evolutionary biologist Richard Dawkins, in his book, The Greatest Show on Earth, says the following: the genetic code is universal, all but identical across animals, plants, fungi, bacteria, archaea, and viruses. Now, based on this observation, Dawkins goes on to conclude that this supports that there is a universal common ancestor. In other words, common descent. Others object and say, well, no, this isn't. This is evidence of a common designer where the same information or pattern is used by a designer. So what is the evidence actually telling us? That's what we're going to discuss tonight. And it's at this time I want to introduce our moderator for the evening. His name is Mark S. Phillips. He's an assistant chapter director of Ratio Christi at Marshall University in Huntington, West Virginia. He teaches undergraduate and graduate apologetics and philosophy courses at Tri-State Bible College in South Point, Ohio. He's a digital designer and publisher. Mark holds a Doctor of Ministry degree in apologetics from Southern Evangelical Seminary, a Master of Divinity in apologetics from Luther Rice College and Seminary, and a Master's of Arts in Religion and Biblical Studies from Liberty University. He's a public speaker and member of the International Society of Christian Apologists and resides in Proctorville, Ohio, and is an all-around nice guy as well. So give Mark a hand as I introduce him.
Boy, with a build-up like that, I don't know. Well, thank you all for having us here tonight. I appreciate the the pastors and the staff here at England Hill for having us in. Not a lot of local churches would do this, quite frankly. This is a topic where a lot of good people get very scared at the idea of actually getting in here and having a cordial discussion about things. And I thank the church for doing that. That's a wonderful thing. I'd like to thank Chan for his enthusiasm through all this. It has been rather infectious. I'd like to thank the Secular Student Alliance. I'd like to thank Rob Fuller for extending the invitation for me to be moderator tonight. And of course, I'd like to thank Drs. Mays and Jackson for their time tonight as well, if you wouldn't mind giving them a round of applause here. Okay. Right. Now, I have a personal vested interest in this topic. At my own church in Proctorville at First Baptist, I teach what's known as a high school apologetics class for 9th graders to 12th graders. And then with my work at students at Marshall University, their ages can range up into the mid-20s. So I have a 10-year window of what everyone from high school freshmen to college graduate students are thinking about. And this is one of the issues that in my discussions with students who've kind of walked away from their faith, it's usually over issues of science. And when they were members of a local church, such as this body, sometimes they felt their questions weren't heard, or perhaps they were dismissed without being answered at least as openly and honestly as possible. So this is a good thing tonight. So I have 3 rather interesting windows onto all this. And if you see me taking notes, it's merely because I need to be able to communicate this information in 3 different places: at my own church, on a secular college campus, and then on a Bible college campus. So there's a wide range of audiences there, and I want to make sure I communicate what these gentlemen have to say as accurately and honestly as possible. So without any further ado, let me describe our format tonight for the debate. Each of these gentlemen gets a 20-minute opening statement. Then each participant gets a 6-minute rebuttal. Then we begin an interesting back and forth discussion of 30 minutes here at the table, at which I'll scoot my seat back a little bit. And each gentleman will be questioned for a 5-minute period, back and forth until 30 minutes have questioned by the other for 5 minutes at a time. It'll go back and forth, and it will be this is a cordial evening, and we do feel very welcome here, and we thank you for that. Then there'll be a 5-minute closing statement from each gentleman, and then there'll be a question and answer session following, and as I understand it, we'll have a microphone set up, 2 microphones, one for each of Dr. Mays and Jackson. So, to introduce our speakers tonight, there was a coin toss. Dr. Mays will go first, but let me read the bios of both gentlemen so you know their backgrounds before we begin. Dr. Charles Jackson, who is seated on your right, has 2 master's degrees in science and education. He has a Doctor of Science Education degree from the University of Virginia. He's taught 15 years in secondary school science, 15 years teaching college sciences, and is an established speaker on topics such as this, especially this of interest tonight, in creation science education. And so if you're interested in reviewing more of his materials, he has a Facebook page titled Points of Origins. Dr. Mays, who is seated on your left, has a bachelor's degree in biology and anthropology and a PhD in biological sciences from UK. Oh, come on, I'm in Kentucky. All right. Yeah, all right, yeah. He was a 2-time National Science Foundation postdoctoral fellow at Auburn University, and that was following graduate school at UK. He's taught general biology, genetics, and evolutionary biology at Georgia Southern, Thomas More College, Xavier University, and the University of Cincinnati. Currently, he's an assistant professor in the Department of Biological Sciences at Marshall. He has nearly a quarter century of collaborative research studying animal behavior, population genetics, molecular phylogenetics, and phylogeography and speciation that has taken him to conduct field research on 3 different continents in the island of Hawaii. He gets to go to Hawaii for work. Think about that for a minute. Yeah. Dr. Mays and his colleagues have nearly 30 published review articles in peer-reviewed scientific literature on everything from the mating behaviors of birds to species delimitation in birds of East Asia to the genome sequence of the Sumatran rhinoceros. He has participated in active public research to spread science literacy, provide career growth for students in the life sciences, and foster a public appreciation for the natural world. And you can find his work at www.monophilia.org. All right, well, thank you folks very much. And would you please welcome Dr. Herman Mays.
Got it? Oh my gosh. Well, howdy, y'all. I'm really glad to be here. Thanks. I just thanking all the people that brought me here, and especially thanks to y'all, the congregation of England Hill here for having me. I really appreciate it. So, a little bit about me. I grew up in Kentucky. My family is from Logan County, West Virginia. Most of them. We've lived there since before there was a West Virginia, and the rest of my family, a smattering of them, lived in eastern Kentucky. So down the road in Moorhead is my dad's family farm. I've got cousins from Stafford that grew up in Staffordsville. And most of my family's been in the coal industry until me and my cousins. So now we have a biologist and a FBI agent and people that work in healthcare in my family, and someone in the country music industry, and lots of different people outside nobody works in the coal industry anymore in my family. So that's a little bit about me. I'm from around here, and like you said, I went to UK, so I grew up in Lexington and lived there we moved there when I was in the first grade, and I finished my PhD in Lexington, so lived there a long time. Now, one thing you know about kids, any of y'all have kids or grandkids, everybody? Yeah? One thing they like is these, right? Anybody have a grandson or granddaughter that likes dinosaurs? Yep. Me too. So I loved dinosaurs when I was little. And that translated into a love of pretty much any kind of critter that I could find. And that expanded out to everything else, too, about nature. So I grew up with a real fascination of the natural world, and I just wanted to learn everything about it. And I've been blessed enough to have a career in science where I get to do that every day. And it's been really great. And so we go all over, and we, like was said in the introduction, we do genetic work on different animals from around the world. And we study things about their history and their relationships and their behavior and all sorts of things. My son is into it too a little bit, although now lately, since he's gotten a little older, he's 13, he's into sports more. But he's, him and lots of other kids that I work with, we take them to catch birds. I worked at the Cincinnati Museum Center for 7 years before coming to Marshall. We had the kids, high school students there, learn how to do DNA techniques. A lot of them are either in school or graduate school now or jobs in the biotech industry. So one thing I love doing is teaching kids about science and nature. So one thing about our topic here is people disagree on it to some degree, and people disagree on a lot of things these days. And so if you look, here's what Michael Oreskes, who's the head of news at National Public Radio, said there are people who understand that if you can create a different understanding of reality, you can actually change politics or anything else you want to deal with. So what I'm going to try to impart on you today is a little bit about the reality of the science of evolution and common ancestry here. And I know I've got limited time and I've been droning on, so I'm going to kind of skip ahead a little bit. So let's talk for a minute about creationism. What is creationism? So creationism in general, just in a general sense, invokes a divine agent as a creator. Creationism has a creator. Scientific creationism is the sort of creationism that takes that idea and presents itself as a scientific idea. So it tries to mesh this belief in a creator with the scientific evidence. Now, creationism per se differs from scientific creationism. So you can be a creationist and accept evolution. You don't necessarily have to reject evolution to call yourself a creationist, and many people don't. And intelligent design theory, if you've heard of that, is a form of this scientific creationism. And most varieties of creationism include some independent genetic origins of species. So they say that species appeared independent of one another and they didn't share a common ancestor. So what is evolution? So evolution is the idea that life shares a common ancestry and it changes over time. Now what changes over time? And populations change over time in evolution. And it's the fundamental organizing principle in the life sciences. This is a tile on the floor at a major university science building, and it says on it, it's by this geneticist called Theodosius Dobzhansky, and he said, nothing in biology makes sense except in the light of evolution. And when we get to the end of this, if I make it to the end before my 20 minutes is up, I'll tell you where that tile is. So, I'm going to be here for about what? A couple hours tonight? I can't tell you all the evidence for evolution. I can't do it. There's too much. So the evidence for evolution comes from paleontology, geology, anatomy, physiology, genetics, cell biology, developmental biology, zoology, botany. I can go on and on. I just don't have time. You can't, there's so much evidence for evolution, you can't explain it to someone in a 20-minute intro, or even, we could sit here all night and I still would only scratch the surface. I can suggest some good books for you. So if you want to read a little bit about more of what I'm saying and dig a little deeper into this, you can come see me afterwards and I can point you in the direction of these books. And some of them are very quite good and very readable for someone with just, even if you have kind of a basic background in biology. Maybe it's been a while since you've had biology, but they're well written where you can get the ideas. Now, one thing about science that's important is what we call consensus. And so if you've ever heard of Isaac Newton is famous for saying, if I've seen any further, it's by standing on the shoulders of giants. What he means by that is he can't do science unless he builds on what came before. And science in general has to agree on things. And so what we do is we examine the evidence and we argue about it amongst ourselves. And people with expertise look at that evidence and they try to convince their peers. And eventually, decades of doing this, people come to a consensus. The idea that the earth is a sphere is a consensus. The idea that the sun is in the center of the solar system is a consensus. The idea that matter is made of atoms is a consensus position in science. So this is a community endeavor that scientists do to achieve this sort of agreement. And they achieve this agreement based on everybody arguing about the evidence. Now, what's the consensus position on common ancestry? So this idea that life shares a common ancestry and evolves over time is as strong a consensus position in science as you can possibly get. It's as strong a consensus position as the idea that matter is made of atoms. Scientists agree on this. So what about the public? So about between 50 and 65% of Americans that are polled believe that humans evolved. Whether that involves God or not is a separate issue, but about between 50 and 65% of Americans accept that idea to some degree. 98% of scientists surveyed by the American Association for the Advancement of Science agree with that position. So there's a bit of a disjunct between the public's consensus and the scientific consensus on this. So why do people not believe in evolution? Well, there can be lots of reasons. And they can range anywhere from religion, maybe there's just a general sort of, we don't trust these hippie college professor kind of things, and that, that might have good reason for that sometimes. Education, political ideology, And then also being in these insular social environments, what sometimes you hear it's called as an echo chamber. You might not hear about the evidence for evolution from someone such as myself. Now, people who want to create a different narrative story about common ancestry are very good at manufacturing doubt. And so what they can do is they can create this counter-narrative with some alternative facts. There are no alternative facts, there's just facts. But some people say that there are, these other facts that support their story. They can also generate their own experts. They can play the victim or invoke a conspiracy, saying that there's a man behind the curtain somewhere that's manipulating all this and hiding data from us or something. They can appeal to fairness. Say, well, isn't it fair that we only have both ideas out on the table or in the schools? They can attract people to information islands, so attract people to places where there's information that they give about their narrative, but they don't give the rest of the information from the scientific consensus. So these are all ways that people can, certain people can generate doubt about common ancestry while it's such a strong consensus position in science. Now, what's the scientific model? So common ancestry is a scientific model that we have. Models make predictions about the data that we collect. You go into your science textbook when you were a kid, and what did you have? You had a picture of the solar system, right? Or maybe you're lucky enough to have like an actual model of the solar system. What did it look like? Well, there's a sun, there's what's next, Mercury, there's another sort of concentric circle of Venus, and then the Earth and Mars, etc. That is a model that people pose to explain the things that they see in the sky. The things that they saw in the sky happen to match that model. That model explained those observations better than another model. Maybe another model that said, well, it's the Earth that's in the middle, and the sun is going over here. To have the observations match that model, you had to assume a lot of weird stuff. It just didn't fit. So the heliocentric solar system is a model. And we look at the world around us to see if the world matches the predictions of that model. Common ancestry is the same thing. It's a model meant to explain the data, and it makes predictions of the data, and then we go to the data and see if the data matches what the model says. That's how we test it. So the idea is, what we're basically saying is, how should the world appear to us if a particular model is true or not? And that's what we go to look for. So you'll see these kind of diagrams as I'm talking. This is a phylogenetic tree. All a phylogenetic tree is a diagram of relationships between things. This branching pattern, the branch points are called the nodes, and the tips are where you can imagine different organisms would be at the tips. And what this diagram is meant to display is, if you can imagine zooming in on those lines, there would be different pedigrees there. And those pedigrees would be parents and offspring in a population. And at places where that phylogenetic tree splits is where the population splits. But you can imagine these lines are filled with individuals in a population that are all interbreeding and having kids together. So it's a lineage. It represents a lineage of ancestors and descendants over time. The branching pattern on there depicts relationships. So just like your siblings are more related to you than your cousins are related to you, the reason that is because you share a more recent common ancestor with your siblings. That's your parents. The most recent common ancestor you share with your cousins is a grandparent. That's why you're more related to your siblings than you are your cousins. And that sort of relationship is the kind of thing we can display on these trees. They show relationships between different organisms. Here's how we test these trees. We can take the data, so this can be from morphology, from how plants and animals look, to sort of deeper looking into plants and animals and seeing what DNA they have, and comparing that diversity, and then testing, seeing how the data fit our different trees that we could pose. So one way is we can look at, so if you start off what we call the prior probability, so here we have 3 groups of organisms, we'll call them A, B, and C. So there's 3 possible sets of relationships they can have. Now before we collect any data, we say, well, let's just say all those trees are equally probable. And then after we collect the data, we can say, now after we have the data, which tree is more likely, given we have this data now? A likelihood method looks at the probability of the data given a particular tree. So given tree 2, the data that we collect is much more likely. That's why that bar is higher. Another method is what we call Bayesian method. And we can tell us the probability of the tree given the data. They're just two different statistical, sort of almost just statistical philosophies and really doing things. But they have all these different methods associated with them. And that also tells us that tree 2 is more likely given the data. So that's how we can take the data and we can say what tree is best supported by the evidence. So that's how we test these trees. Now, if you look at common ancestry versus common design, common ancestry says different species share the same genetic history. They're all related. Common design, or creationism in general, it's if you adopt a scientific creation version, not all versions of creationism reject common ancestry, says that each one of those species appeared independent of one another and they never shared the same ancestors. So instead of the common ancestor model being like a single bush or a single tree, you can imagine the common creation or the common design model as being like a forest, with each group of species being a bunch of different trees that aren't related. So those are two models. We can take the data and compare the data against those two models and see which one fits the data better. So we can actually test these ideas. Now there's some caveats to make here. So one is universal common ancestry. That says, is everything related? Okay. Another is we can look at common ancestry of specific groups. So we can look at all primates. Are all primates related? Both of those can be tested by the same approaches. So I'll just skip ahead here a little bit because I'm running out of time. And I'll go back. Any of this that you need me to explain more, ask me in the Q&A period and I'll explain it to you. So if we talk about neutral variation, so the variation we're looking at in genetics has nothing to do with variation in proteins. So you can have your genes vary. So two organisms can have different genes, but they make exactly the same protein. So there's no difference in terms of function in that protein, but they have genetic differences. So those differences we call neutral because they don't have any impact on the function of the organism. So when we look at neutral variation, here's an example of a paper where they looked at primate genes. Now these two graphs, and I can explain this again in the Q&A for more detail. It's looking at stretches of DNA that encode for amino acids, and amino acids are little building blocks of proteins. The gray bars on the left is the diversity of codons that we observe if we compare a bunch of data sets from primates, the studies on primates. The black group of bars on the right is the distribution of variation at these genes that we would expect if they were independent. They didn't share a common ancestry. So what we see here is that there's this huge gap between the two. We can test this further. We can take two phylogenetic trees and we can see how similar they are. So one based on amino acids, which would be more a functional tree, and one based on nucleotide sequences or DNA sequences, which would be neutral. We're looking at neutral variation. It doesn't make any difference in terms of the function. If we do that, what we get in this same study, so I'm referencing this same study now, that's about an odds of 1 in 40 billion of getting a match that's that good or better between two trees, two different trees, if they were independent of one another. So again, this is a test that rejects the independent origins model. Here's another one. So this is one based on the sort of the shape of the trees, the phylogenetic trees that we look at. On the right is the distribution of data we should see if common ancestry is true. Sorry, on the left is for common ancestry, and on the right, the black bars, is common design, say, or creationism. They don't match. So again, we reject the common design or the independent history model. Now we can look at specific groups. So here's a bunch of mammals. So there's a whale, a cow, a manatee, and a shark. So if they were created independently, they look like on the left. If they had shared a common ancestry, they should look like a tree on the right. Common design would say all the aquatic critters should be on the same branch and all the land critters should be on another branch because they should be designed the same way to live in the water. So which one of those trees Does the data match? Well, if we go through the evidence, it matches the common ancestry model. And here's one this is from a certain type of DNA called ribosomal RNA, and this places whales nested inside the hoofed mammals. That includes deer and hippos and giraffes and antelope and things like that. What do we get from a completely independent gene? These are casein genes that are in milk. You get the same pattern. It's independent of the other study. What do we get in the next one? This is taking whole genomes of organisms and splitting them apart and sticking them together and see how similar they are. We get the same result. What about if we look at these other genetic pieces of information called transposons, which are little bits of data that jump around the genome? We get the same pattern. Again, independent of the other patterns that we saw. What if we look at a whole bunch of genes? These are genes in your nucleus, nuclear DNA. 26 genes altogether. We get the same pattern. What if we combine nuclear genes and these transposons together? We get the same pattern. All of those bits of data are independent of the others, and they all tell you the same story, and that's not what you would expect based on independent origins. Now, can a designer explain those data? Yes. Actually, yes. So if you pose your designer is an omnipotent being, omnipotent beings can be used to explain absolutely anything. And so there's no way to rule them out. So while you could pose an omnipotent designer to explain those results I just showed you, an omnipotent designer isn't a scientific model because it can explain anything. So I'm just going to go to this real quick. This is what I showed earlier, the tile from Dobzhansky that says nothing in biology makes sense except in the light of evolution. Where is that tile? Does anybody know? So it's on the floor. Yes. So that tile is on the floor of the science building at the University of Notre Dame, a Christian university. So that's it.
All right, thank you, Dr. Mays. I appreciate that. And now we have Dr. Jackson's opening 20-minute statement.
On? Okay, we're good, we're good. So let's take a look at that, the topic tonight. Now this is from the Phi Delta Kappan, which is their top drawer educational journal. Back in 2000, when I read this article by Mano Singham, they only accepted 5% of the manuscripts that were actually submitted. So it's really, really up there. Look what it says here. And this is from an article called The Science and Religion Wars. The vitriolic tactics that have been adopted by both sides in the science and religion wars seem more likely to alienate people than to win them over. And I think that's true. There's a lot of mudslinging. No one's really attracted to either of the things that are being suggested. But take a look at what he says after this, and I think it's very important to what we're doing tonight. And so far, in the spirit of everything, it looks good to me. That would be a pity, because the issues raised are deeply interesting and have profound implications for anyone who seeks an understanding and a synthesis of science, religion, and philosophy. I presented this at the East Tennessee Math and Science Summit at the University of Tennessee, Knoxville, to a bunch of science teachers in Knox County, and really got a lot of good discussion going. Good discussion, good dialogue, good discourse is what cranks the grist of science and the science community. Linus Pauling said, "What is science? It's a search for the truth. the effort to understand the world. Science should be a search for the truth. Ultimate truths, whatever, comparative truths, it's a search for the truth. Religion should be a search for the truth, for ultimate truths. These used to be sisters, and we're going for the same thing. Isaac Newton said, all of science is just thinking God's thoughts after him. So that kind of a worldview motivating the search for the truth. And Richard Feynman, also a Nobel Prize winner, says science is a long history of learning how not to fool ourselves. How can we fool ourselves in science if we, and you were talking, if we're vested in some kind of a brand name thing from the start? So I think, how many people know that there are in the church has some hypocrites hanging around in the church? Okay. You know, there are also some hypocrites hanging around in the world of science too. Both of them maybe aren't, and some of the members of these groups might not really be looking for truth. They might be looking to prove something they hope is true or want to be true. And so what we have to do is just drop all the pretensions and look at evidence. Evidence is not proof. Evidence is not proof. Everybody has the same evidence, the data. It's just different interpretations. How did it get this way? So that was my intro. Let's see, I guess I should be able to finish. Okay, here we go. Louis Pasteur, in 1864, announced to the French Academy of Sciences that life comes only from life. That the biogenetic, or the biogenetic law, and he said, do not put forward anything you cannot prove by experimentation. And of course, you can't experiment very much with evolution and creation, but you can look for evidence after the fact. This is Darwin's little notes that we found that he left, and this is his first little rough sketch of the common ancestry tree. Darwin himself said, a fair result can be obtained only by fully stating and balancing the facts and arguments on both sides of each question. And this is here impossible. Well, it's been over 150 years. Well, now maybe we can. There's a lot more data that's come in, a lot more information, a lot more thinking that's been put into this since the time when Darwin wrote his book. Let's take a look at some of the assumptions that descent makes, the descent model, and the design model makes. And then look at some of the predictions that you would expect. And I want to do a compare and contrast on these assumptions leading to these predictions. For descent, one, you'd expect the phenotypes, in other words, what things look like, to parallel the karyotype, what the chromosomes look like, and the genotype, what the DNA is like, what the genes are like. And I want you to notice that, as Dr. Mays mentioned too, phenotypes paralleling the way that the way things look, paralleling the way that the chromosomes look, the way that the genes, the DNA and the genes are, should be the same for whether we were designed or whether we were of common descent. Actually, all of our DNA should actually be more similar with creatures that look more similar to us, that have more similar proteins, because the DNA is coding for proteins. So this is something that's similar between them. And then mutagenesis, mutations, actually generating new genotypes, new DNA, new genes. For bacteria to turn into dinosaurs, you'd need that to happen. Or then phenotypes, the phenotypes, again, just what they would look like. And what you'd expect to see is some kind of evidence that you could find fossil DNA, fossil genes, leftover genes that aren't used anymore, things that are basically fossilized. They're in there, in the DNA, and they aren't being used. They're neutral to the entire situation of natural selection. It's not going to kill you if anything goes wrong with that, because we're not using that DNA anyway. And it should be free, because natural selection doesn't affect it, to mutate into all kinds of different things and never hurt anything or help anything. And just sit there and be very, very different over a long period of time. For the design assumption, we'd think that the mutagenesis would just not work because of the beauty and the complexity of the design. If you messed with it, you really wouldn't do anything that would help it. If anything, it would hurt it. Not to say that mutations haven't hurt the genome over time, but that you couldn't actually get anything to become something better. And then the last thing, this neutral selection thing, which I just mentioned, I separated it in this way because I want to show you some of the data that has come about that separates into these areas easily. Neutral selection, you would get junk structures, vestigial organs, like I say, are some organs we don't use anymore, they don't have any function, and then vestigial DNA. DNA that we don't use anymore, and it doesn't have any function. It's lost its function over time, and there's only certain DNA that we actually use, since we're not worms and fish and lizards anymore. And then the design assumption would be that there wouldn't be any useless organs. There might be organs that don't work as well or don't work anymore, but none that don't have any use. There might be some DNA that might have been damaged, but nothing that's just this huge amount of stuff that never did, what doesn't have any use for us today. It might be damaged. A few DNA genes might be damaged but by mutation, but not that they don't have a use. Very, very little of it would be like that. So let's take a look at that first one. And I'll take the positive because this is something that both the descent model and the design model would both assume would happen. I take issue with that picture because the chimp's head is really much smaller than that. But why do we look this different? We actually are very similar, chimps and humans, compared to other things. Now this is just comparing the karyotype. What the chromosomes, little shoestring things in the nucleus look like. The karyotype and the genotype, what those little strips of color actually say in the DNA, are both things that scientists look at when comparing. And it's true, orangutan, gorilla, chimp, and human, they do look very similar. What would expect that since our bodies look similar, our physiology is similar, and our biochemistry is similar. That would be true whether design or descent was true. I like this picture better. At least it has a chimp next to a baby human's head, which is a little bit more reasonable on that. We do have a lot of DNA that is the same with chimpanzees. The article there in the magazine said 1.23% difference. But out of 3 billion, that's still 30 million base pairs different. But still, why do we even look that similar? 98% of our DNA being the same, we should look a little bit different, but maybe not that much. What's the explanation? Let's take some perspective. The nematode worm has 75% the same DNA as us. It only has 1,000 cells in its body, 1/10 of a millimeter. That's just showing that all living things do have very similar DNA. Bananas have 50% the same DNA as us. All of the different things for all the cells to work and for the bodies to put together, even in plants, would have a lot of similarities. So why do we look as different as we do from chimpanzees? We would expect, since we look like them, though, that the DNA would be similar. But it's so similar. There was some more scientific research that went on. Svante Pavo at the Max Planck Institute got a runner-up award in 2006 for figuring out some of this. He did the Neanderthal genome. But look at what he says. He found, well, it had been known, the FOXP2 gene, which is a developmental gene. It has to do with the development of the brain. And it's in lots of mammals, not just humans. But it's also in chimps. And he said that the FOXP2 gene produces a subtly different protein in humans with only a difference of just 2 amino acids. Now, most of your proteins and enzymes have got, ballpark figure, 500 amino acids. So having only 2 different, how could that make a difference in brain development where their brains are 1/3 the size of ours? Well, what he found out was that the gene activity was very different. Gene sequences don't tell the whole story. He found that many genes, around 10%, from those studied, expressed at significantly different levels, different amounts. So they were cranking out the same protein, but cranking out a lot more. So difference in gene activity, how much is it releasing, how much of this protein is it manufacturing, is one of the things. But how does it know is one of the mysteries. And so we went a little further in the research on this. Let's take a look at the descent assumption. Again, with the mutagenesis, making new genes and new looking creatures, we should find fossil DNA, DNA that's no longer good in living things today, that isn't being used, like junk DNA, and fossil phenotypes, in other words, missing link skeletons and fossils like that. We're not going to talk about that much today, but that's a prediction of the model also. And natural selection. would leave those junk structures alone. They would continue to persist, and so would the junk DNA. Okay, this was a very big mantra in the descent theorist community for many decades in all the textbooks. How many of you ever heard of junk DNA? Yeah. How many of you heard that it's been disproven? How many of you have heard that it was proof for evolution? Yes. It'll be in the books, and it takes a long time to get these kind of things out of the books, but let's take a look at what happened. 2005, this statement, a lot of the DNA in there is not needed, it's junk. If it's intelligently designed, then God needs to go back to school, a professor at Columbia University. So let's take a look at, and I'm just putting that back up so you can see that again, but let's look at what has happened since 2005. Also, Richard Sternberg, who was in the big hullabaloo about allowing Stephen Meyer's article to be printed in a journal that came out of the Smithsonian, that's the Smithsonian behind him, said this with James Shapiro in 2005 about junk DNA. One day we will think of what used to be called junk DNA as a critical component of truly expert cellular control regimes. In other words, we're going to find out there's no such thing as junk DNA, and that was a prediction of the design model. So let's take a look at what has happened since then. And it's not just complicated. You know, the design model says it's very, very complicated. This is a very simple thing, easy to understand, that maybe if one gene gets mutated, it's going to affect a whole lot, because a master control gene, a regulatory gene, is going to tell another gene what to do, which might tell another gene what to do. It'll tell this one, that one tells this one, tells this one, tells this one. So you mutate one of these, and this is one of the theories that's saying, well, this is how the big changes could happen. But if you mutate one of these, you're going to affect the activity of many, many different genes. So it's a little bit nested within nested, and it gets a little bit complicated. If you mess with a perfect system like this, you're going to mess with a whole lot of things. Now, 2 years later, then, in 2007, 29 papers from 11 countries found out that the junk DNA really isn't junk. So this was a prediction made by the design model that this would be true. And now it's really surprised everybody. Most of a cell's DNA gets transcribed. In other words, it's used, it's put into action. The expectation was that most active DNA sequences, well, that has more to do with lineages. I'm going to try to stay on time here. 93% of the genome is transcribed, not 3% like predicted by the descent model. And this is not from the Creation Journal, it's from Nature magazine, one of the most revered journals of science in the world. Further study with more wide-ranging methods may raise this figure to 100%, that all the DNA is used. There's no junk at all. This was the year 2007. One exon can be used in combination with up to 33 different genes. An exon is a gene that's doing some making of proteins. And it's located on 14 different chromosomes. So one gene can actually affect a lot of other genes. They don't even have to be on the same chromosome. This means that one exon, yes, can specify one part shared in common by many different proteins. There's no beads on a string linear arrangement of the genes, but rather an interleaved structure of overlapping segments, typically 5, 7, 9, or more transcripts coming from one gene. Okay. See, transcription proceeds not just one way, but both ways, frontwards and backwards. Not just one strand of the two strands on the ladder, but both strands, what we used to call the antisense strand, actually gets read and the information is used. Sometimes we call some of this epigenetic because it's stuff that's beyond the genes. Okay, let me see. Then in 2012, this is what happened. More research came through. The shadowy part of the human genome, once disparaged as junk DNA, a view radically at odds with what biologists have thought for the past 3 decades because of the descent model. 97% of the human genome that's not, strictly speaking, made up of genes. 97%, we didn't know what it was doing. We figured we're so smart, if we can't figure out what it's doing, it must not be doing anything. So it's just junk as predicted by the descent model. The Encyclopedia of DNA Elements Project, nicknamed ENCODE, identified 4 million sites involving regulating gene activity. Instead of actually making proteins, they're telling all the other genes when to make proteins. That's why it was so hard to find these. Previously, only a few thousand of such sites were known. In all, at least 80% of the genome appears to be active at least sometime in our lives. 80%, not 2%, not 3%, which was a prediction of the design model. Further research may reveal that virtually all the DNA passed down from generation to generation has been kept for a reason. That kept for a reason is not a design, but a descent phraseology on this. Look at what it says here. This concept of junk DNA is really not accurate. It's an outdated metaphor, says Richard Myers of the Hudson Alpha Institute for Biotechnology in Alabama. The New York Times that same year reported more on this very same wave of research that came through in 2012. At least 4 million gene switches that reside in bits of DNA that once were dismissed as junk, but that turn out to play critical roles. That's what Sternberg said would happen someday, 7 years before that, in controlling how cells, organs, and other tissues behave. As scientists delved into the junk, parts of the DNA that are not actual genes containing instruction for proteins, they discovered a complex system that controls genes. Eric Landers, well known in the descent community, president of the Broad Institute, working with Harvard and MIT, said, "My head explodes at the amount of data. Human DNA is a lot more active than we expected, and there are a lot more things happening than we expected." And then Ewan Burney, also a big name in the scientific community, lead researcher, and he said that. And that Dr. Bradley Bernstein, an ENCODE researcher, said it's a really big deal. He added, "I don't think anyone predicted this would be the case," which I take exception with because the design model did predict it. We've been saying this for decades, that there's no such thing as junk DNA. There wouldn't be junk DNA. Sure, there might be some damaged genes that suffered damage from mutations over the years, but that the majority of it, just a little bit of it, doesn't work anymore, would have had a purpose from the beginning and always would, and wouldn't be a throwback or evidence of throwbackness back to when we were worms, fish, or lizards, or anything like that, as the common descent model says we once were. So that's just a summary there of those assumptions that are made and the predictions that those assumptions lead to, and then what I just showed you again. As Dr. Mays said, there's no way to go over this whole topic in 20 minutes, but showing you a segment, maybe an example of how deep the rabbit hole can go, and if we had a week to do a seminar, we could probably touch on a lot more different things that are pertinent and have bearing on the contrast between the two models of descent and design. Thank you.
All right, thank you, Dr. Jackson. Now we will enter into a rebuttal period. And Dr. Mays, do you need a few moments, or are you ready to?
No, I'm ready.
Okay, if you're ready,
Sure.
we're all set.
Can I borrow the
Yes,
thing here?
All right, thank you.
Can you switch to my slides? Thanks. Junk DNA is a popular topic amongst critics of evolution, creationist critics among evolution. And I had a little extra slide here because I kind of anticipated it would come up. So here's the ENCODE project he was talking about, is the first line here. There's big RNA. So you know there's DNA, and it's a molecule, and it gets copied into another molecule called RNA, right? Does it sound vaguely familiar? You think back to your biology class. Well, there are these long non-coding RNAs. So these are the big RNAs that get transcribed, what Charles said, from your genome. About 80 to 98% of those RNAs identified by ENCODE are present at only one copy per cell. Okay, so they're present at such, when you look at RNAs that are doing something in the cell, they're there at very high copy numbers. Being at such low copy numbers is evidence that despite that there's transcription, there's not a function going on in the cell. And then also, if you take RNA polymerases, these are the enzymes that copy RNA from DNA. They're prone to transcriptional noise. So another study here, this is the Stroll study in 2007, said that 90% of yeast RNA polymerase II events are noise. And in fact, if you take RNA polymerase and you put it in a test tube with random bits of DNA, it will transcribe things. And I don't think there's any function there in transcribing a random piece of DNA in a test tube, but it will do it. Now, also, mutational load. So mutational load is this idea from population genetics. So if you apply this to look at the genome, if the entire human genome was functional and virtually all mutation was deleterious here, it would accumulate so many deleterious mutations that for the population to remain constant, for a modest mutation rate, you would have to have, everybody in the population would have to have, on average, 466 kids. So that model says, based on the mutational load, that if the entire genome was functional, everybody in the population, to keep the population constant, would have to have over 400 kids. It gets higher as you change the mutation rate. Large non-coding intervals, so this is an interesting one. So this is an experience by Nobrega in 2004 in Nature. They took mice and they took big regions of DNA. One region is 1.5 million bases, and another one is 0.8 million bases, and they removed them. So if the entire genome is functional, what's going to happen when you remove a million, actually in these two things, a million and a half or bases from two different areas? What do you think is going to happen if the entire genome is functional? And you take out a giant chunk. Mouse won't live, right? They do just fine. So you can take out those big chunks, and they do fine. And in fact, many of us here in this room have copy number variation. So we don't have the same, all of us in this room don't have the same genome size. Some of us are missing big chunks that make no difference in terms of your survival. Now also, if you look at the genomes of other organisms, so this is the Vizcacha rat from the Andes. Its genome is 3 times larger than the human genome. So if the entire genome is functional, why does a rat in the Andes need 3 times more genome than you do? Now, not only is it 3 times bigger than your genome, it's twice as big as another rat in the same family that it's closely related to. The flowering plant, this flowering plant, Paris japonica from Japan, is 50 times your genome. So it's just a little weedy kind of flowering plant. Why does it need 50 times more DNA than you do if all that DNA is functional? There are some amoebas. So you guys remember amoebas, like, I love like old science fiction movies. You know the old science fiction movies, the blob that ate Cleveland, you know? So it's kind of, amoebas kind of like that. It's this amorphous single-celled organism that can go around and change its shape, and it gobbles up bacteria all around it. Its genome, it's all this DNA, its genome, is 100 times bigger than your genome. And you're a multicellular organism with, we have brains and stomachs and arms and legs, and we go to college, and we do all this kind of cool stuff. Yet we do it with 100 times less the genome of an amoeba. Now, one thing about junk DNA that creationists always say is that junk DNA is a sort of proof, like he said, it's proof of, it's not. It has nothing to do with the evidence for common ancestry. We can look at neutral variation, and neutral variation is a very good way to test common ancestry, but we don't need there to be junk DNA for us to test common ancestry. So, there, all that. I'll yield the rest of my time. Oh, it's perfect anyway.
Okay, for rebuttal, I wanted to point out the idea of consensus making truth is not always reliable in anything, especially even in the world of science. For 1,500 years, the geocentric, the Earth's in the center and the sun goes around it model was the consensus. I wonder if it was 2% of the scientists that disagreed with that consensus in those days. Also, the idea of creating doubt. I know that there might be people out there doing that to try to push the design model. But alternative facts, well, I guess another word, I'd agree with that, with Dr. Mays. Alternative fact is lie, isn't it? Or an untruth. but there are alternative interpretations of the same facts, the same data, the same evidence that you look at, not pretending that something isn't there or pretending something is there that really isn't, but interpreting how did this get here? What's the backstory? Just like a crime scene, we're looking for the backstory. So interpretations that different models would have for what we actually see that ended up in the end. That, I think, is the thing. Anybody who is trying to create doubt with alternative facts, well, they're not in my camp. They'd be in somebody else's. I don't know whose. And the idea that the design theory says that every species was created independently, in design theory, it's not every single species, but the definition of species has gotten very blurred over a long period of time. You know, Canis lupus, the timber wolf, and Canis familiaris, the dog. You know, you can have a wolf dog. You can breed them together. That we give them two separate species names, but they're really not. According to Carolus Linnaeus, who created the binomial nomenclature system and taxonomy of living things, it was things that could reproduce with each other. Coyotes and dingoes and dogs and wolves can all interbreed with each other. So, for the design model, they would be the same kind, or in the Hebrew word, bara min, or created kind. And then all these different, they're really varieties of the same thing. They get off and breed for a long time and get a lot of mutations that can't backbreed to the old population. You might call it speciation. But, so what I'm trying to say is, it's not all this, every single different species was exactly the same, but rather that these groups that are really related, yes, came out from the original created kinds. We used to call it the creationary lawn, and we called it the evolution phylogenetic tree, but really, the design theorists today call it the phylogenetic orchard. For instance, when turtles were first being put through this kind of thing, there's a scientist named Wayne Frere, who I've actually gotten to meet, a wonderful man and also a great scientist. Back in the 50s, he did the blood serum analysis, analyzed the proteins in turtle blood, trying to figure out how many groups of turtles there really are. And he did all this research, and then he did it again when what you would call electrogelphoresis, but DNA fingerprinting came in, and we were able to even nail it down more. And he found that there were 4 main groups of turtles, and they really could have come from 4 created turtles, 4 initially designed turtles. The long-necked water turtles, the short-necked water turtles, and the tortoises and the turtles. And so all living turtles could have come from just 4 originally designed turtle kinds, and then branched out into what really are varieties. And we could call them species, fine, but the designed kinds would be more like the level of genus or family, like the dog family or something like that, the Canidae. So these groups are not, it's not necessarily all the different species or what we're calling species, but it is all of the original design kinds that then did not mutate, although mutations could have occurred, but then these varieties sprung up from them, like the 200 different breeds of dogs that we have today. Also, the pattern that we see and the similarities in DNA, I did kind of go through that at the beginning there. Yes, design explains it. Yes, definitely descent would explain it too. But these similarities, it's not that, that it's a one-size-fits-all thing, or both of these would have to be called that. It's that these similarities can be explained by either model. It's the similarities in design would be showing the very same evidence. We'd see the same patterns, and there are some other statistical things that he brought up, but the overt patterns would be the same. And that's, I got an extra minute, but I don't need it either. Okay. Thank you.
All right, thank you, gentlemen, for your initial presentations and rebuttals. Now we'll take a moment where, take just a few moments to gather your thoughts and a few questions. And since Dr. Mays went first tonight, we'll, if it's all right with you, Dr. Jackson, you he will ask you questions first, if that's all right. That's my decision, and I'm sticking to it. So, all right, gentlemen. Remember, we all love you, each of you. So please. All right. Let's, and I will speak again at 30 seconds to let you know when this period finishes. So let's go.
Who's asking first? Me. I'm asking him first. So what predictions would your model make for neutral variation? So say third position codon variation at protein coding genes, and especially comparing that to an amino acid tree.
Are you talking about redundant codons?
Yeah, degenerate, the third position.
Well, in the first place, we're finding that they're not really all redundant. A different codon that could code for the same amino acid actually might transcribe faster, might synthesize the protein faster, which might actually make the bend in the folded proteins a little different. So we would be tending more, I think the design model would tend more towards there's probably more complexity to this than meets the eye, and that the redundancy could be nice to make sure that mutations don't affect anything and that it really truly is neutral. But as you probably know, the new research is showing even the redundancy may be something that's not as true as we thought it was, and not just methylation and things like that. Does that answer the question?
No.
Oh, okay. What would the design model say about it?
What would you
the
predict
neutrality, neutral to natural selection?
First of all, on the codon bias thing that you brought up, at what level is the bias? Does the research say the bias exists?
It's new, just the past year or two that I've been reading about it. As far as what level the bias exists, are you talking about the different effects of different codons, of which we considered redundant in the past? Is that what you mean?
No.
Okay.
At what level? Where is this bias in nature? What groups are biased towards what?
By groups, what do you mean? You don't mean species. You mean
Any kind of taxonomic group. I mean, I'm just asking you to tell me what the literature says on this topic in terms of what groups have this bias.
what, are you talking about taxonomic groups?
Yeah. Where's the bias in the codons?
What do you mean by bias in the codons?
Well, you brought it up. You said
okay.
that.
Oh, well, no, I was just talking about in general, in the entire
I know, and I wanted you to be
code.
specific.
Well, it would be across all species that are using the same genetic language. They're still using the same code, yeah. That it's doing that transcription and synthesis that the codons are not as redundant as we thought. Now, in keeping with design, we would say that, yes, the redundancy has a purpose. It isn't an accident. It's not something that happened randomly, but that it was there on purpose. Until this new stuff started coming out, that maybe the codes aren't as redundant as we thought in all living things. We would have just said, you know, like the descent people would say, that this was to prevent mutations from always having a deleterious effect, from always changing the next amino acid that goes on the protein. And now, just again, I think it's a similarity with descent, with the descent model, is that we're seeing it's more complex. Again, like the junk DNA design would say, that this complexity is a part of the design, showing that it's, well, intelligent. Does that help?
Not really, but
Oh.
yeah. But
Well,
I guess
the answer is we like it.
do we have markers for the board? Can I have a marker? Sure. So codons are 3 letters of DNA. You remember DNA from high school is G's, A's, T's, and C's all strung together, right? So, for instance, the amino acid valine, we'll just abbreviate VAL. You can get valine if you have GTT, GTA, GTC, or GTG. Okay? All of those can make valine. So this variation doesn't matter. So if you build a tree based on a phylogenetic tree based off this information, that is neutral because it's making the same
30 seconds.
It's making the same amino acid. This is the bias in mice. So 17% are that, 12% are that, about 25 are that, and about, hopefully my numbers add up, 46 are that. This is in humans.
Okay, that's time right there.
12. Sorry. So there's bias, but it doesn't differ between mice and humans. And in fact, that bias is common in all animals, so it doesn't affect the trees you would build within mammals.
All right, 5 minutes starting now.
Okay. I'm going to assume that you're familiar with the idea that's been touted in the descent community about the simian 12-13 fusion for human chromosome 2.
Yes.
Okay. Could you go ahead and explain what that significance is to the descent model?
So humans and chimpanzees have different numbers of chromosomes. And chromosomes in living things do lots of different stuff. And we see this in extant populations. Chromosomes can fuse together end to end, or they can break apart. And so you can get changes in chromosome number based on these events. It's sort of a type of mutation. So one way we can explain that there's a different number of chromosomes in chimps and humans is that 2 of the chromosomes in chimps that are normally apart are stuck together to make 1 chromosome in the human. And we can tell this, that makes predictions. So what we would predict to see, the ends of chromosomes look different than the rest of the chromosomes. So the ends of chromosomes have these little structures called telomeres. Telomeres are kind of like the little, these little, that has a name. Aglets, is that it? Aglet, on the end of your shoelaces for your sneakers. And if those ever get worn off, what happens to your laces? They just fray and come apart, right? Telomeres are like that for chromosomes because chromosomes don't get copied very well at the ends. So what you expect to see is if there's a fusion between 2 chromosomes, you expect to see something that looks like a telomere in the middle. And so that's what we find. So that's what he's going to talk about.
The theory goes that because simians have 24 pairs of chromosomes and humans have 23, that if you actually look at a picture, can you pull up on at the end of my presentation where it says simian 12-13? Let's go ahead and pull up that blue one at the end. It says simian 12-13. And there's a little chart right after that shows a picture of the chromosome that we're talking about. Yeah, just one more. Just click it. There we go. Okay, so the image of human and chimp chromosomes, there's their 12 and 13 going together to make the human chromosome 2. It actually looks a lot like you glued together simian chromosome 12 and 13. Now, there are some problems with that. These red areas here should be like 15 to 20,000 base pairs, and if there's 2 of them, we're talking about maybe 30,000 base pairs. And really, in this chromosome, there's only 700. But give the descent community, giving them just that this fusion did happen, that humans and chimps did originally have the same number of chromosomes, and a fusion did happen, and now humans only have 23 chromosomes, and chimpanzees but they still have 24. Now, the question is, and I guess I should ask this of you, is when did the fusion happen with regard to what you were talking about, the tips and the nodes? This is the node, and so when would the fusion have happened and then been inherited by all generations after that?
Well, it would have happened in the lineage that led to modern humans.
Okay. Would it, with regard to the node, would it happen before the node or after the node or right at the node?
Oh, no. Probably after, because it's in the lineage that leads to humans. So we don't know the ancestral karyotype necessarily, but you can infer that there was a mutation that happened on the line that led to humans that had that. By the way, we still see these mutations in human populations today. There are people that walk around right now with those mutations, so it's not like they don't occur. They occur.
Yeah. The idea of common descent for this, though, if no simians have the fusion and all humans do, then that would mean that either all the simians that had it before all died out, or all the humans that didn't have it before all died out. The fusion would have had to happen right
30
at
seconds.
the node, whatever the node is, and the odds of it happening exactly at that moment are kind of unlikely when you think of the odds and think of the statistics. Since it is something that actually sets humans apart from simians, it's actually evidence against common descent.
That's absolutely not true. So if you remember my diagram I showed of a phylogenetic tree, and if you imagine zooming in on the branches, in fact,
We'll go ahead and start with your 5 minutes. Go ahead.
if you can switch to my slides real quick.
I'm going to pause my timer just for a moment.
Oh, I'm sorry. I
while
don't
you
mean
get
to
that slide up.
throw you off track. If you remember my little diagram that shows how you read a phylogenetic tree, you think about the branches as being these collections of, it's like all these family lines. It's like everybody went to Ancestry.com and put in their family lineages in there, the whole population. So if you imagine what those lines are, they're just a diagram. But what they represent are unbroken lineages of ancestors and descendants. So see all those squares and circles? Those are males and females. The squares are males, the circles are females. That mutations of chromosomal rearrangements occur right now. There are people in populations right now who have chromosomal rearrangements. So that chromosomal rearrangement he's talking about didn't have to occur here. It can occur anywhere in this line that led to us. So any one of those squares or circles could have had that mutation and then passed on. And so there's all kinds of reasons why you get passed. In fact, whenever you have mutations, they eventually drift to fixation, especially in small populations. So that mutation that occurred somewhere in one of those O's or squares went on to be in everybody. Right? So that's just simply untrue that common ancestry or evolution says that this had to happen there. Nobody says that. So do I still have time? I have 3 minutes. Oh, my gosh. So back to this question, while we're on my slide there, how do you explain this? How do you explain that? So the gray distribution, that's 50 independent data sets where there's 16 families, and they took a different sequence randomly from a species in one of those families, a primate family. So this is all primates. And they made a phylogenetic tree from that data. Now, the gray, the light gray on the left, is the distribution of all those data sets. And it's looking at how variable the codons were at those data sets. The black bars are what you would expect based on 3 different models. And each one of those models, remember you talked about codon bias before. Each one of those models is a different codon bias model. The black bars are what you would expect if different families appeared independent of one another. Most are 100 or at least a couple hundred standard deviations away from each other. I don't know if you guys ever remember statistics, but there's p-values. So a p-value tells you the difference between 2 groups. The p-values, it's kind of like the probability that you would get a match that's that bad or worse. Okay? Those probabilities are in the tens of billions that you would get a match that bad or worse just by random chance. So what it's saying is it's the 1 in tens of billions that you would get a difference between the expectation of common ancestry versus the expectation of common independent genetic histories, let's say. So how would you explain that, Charles?
Well, it depends on how the expected outcome was calculated. This is almost like the letting the fox guard the hen house. is this generated the same way as the sort of the straw man argument about the common design model saying that all species are separate and that they should be far more unique from each other than they really are based on the Berriman idea in common descent?
So here's your
Common
Berrimans.
design.
Here's your Berrimans. Here's your independent ones. They never shared a common ancestry. And so that means that the codon variation that appears in them, it's independent. It's independent of any function. It doesn't make any difference in terms of function. And you've already accounted for the bias problem because they take that into account. How does that neutral variation stack up here? So this says, okay, what's the probability we get a particular valine codon here versus here? Well, that just depends on mutation, right? So any variation you would expect from the creation here would just accumulate through mutation. So if that's your model, you would get those black bars there. So how do you get the difference between those results?
Well, since we kind of went back and forth that way, I'll take some of the time. This is not actually comparing the genes themselves and the traits that come through the genes.
It is.
What you're talking about is, as you said, a bias towards which of the redundant codons is the species, that genome, going to actually use for making the next amino acid in the protein. And so this is, it's not really comparing the genes and the structures. It's not really comparing the genotype and the phenotype. It's comparing the activities in protein, in transcription of the RNA off of the DNA, and then in translation into proteins out in the cell on the endoplasmic reticulum. So it's a functioning aspect of producing the proteins and not actually something of the information and the actual protein that's formed. It's not right.
That's not right. It's the variation that these studies are looking at is independent of function. So the purpose of this study is to uncouple this idea of, well, maybe you get these patterns because there's a common function involved. When you just look at the neutral variation, you've removed that possibility because they're all producing the same thing.
Well, you're calling it neutral because it does produce the same thing, right?
Yeah, they're indistinguishable from one another. And
so
you account for the codon bias problem in those models. So they accounted for codon bias in 3 different methods.
So this is like playing a card game with, and there are 4 cards in the deck that will actually do the exact same thing for you. Maybe it's a card game that only counts on the numbers and doesn't matter with the suit. So what you're saying that suit is neutral. It doesn't matter what suit we're getting. We just need the number, right?
No.
Okay. What are you saying then? I mean, you're saying that.
Well, I'd rather not make kind of an analogy that's irrelevant to the point. It has to do with variation in those codons.
And which one is used the most often to
But you
get
already
the
account
same
for that.
protein.
So for instance, if you go back to my codon bias thing in there, you would say that, okay, the probability that this lineage will have this codon will occur based on its probability that it occurs, right? And some codons it's biased. Some are more common than others. So you just bias your results to say, well, this one should be more common because we know there's codon bias in mice and mammals and humans. So we'll say it should occur at a probability of 47%, right? So that would be the probability that that lineage would get that codon. The next one would get another codon at some, you'd say, okay, the probability it would get the same codon would be 0.47, right? The probability that they both would get the same codon would be 0.47 times 0.47. Right? Now, you do that for lots and lots, hundreds of thousands. In fact, they looked at over 3,000 amino acids here. The probability that they produce a pattern that is similar and they vary less becomes very small because of that.
So there are 4 different codons that can be used for the same amino acid. And what you're looking at is the preponderance in different species of which of the 4 codons do they choose or do they use more often, right?
It's which ones they have. It's the distribution of variation across those different
In other words,
species.
like you said, a bias towards one of the 4 options for getting the same
You already
result.
know there's a bias, and you're just building an expectation saying, okay, we know there's a bias, so given we know that there's this bias, what should the distribution of codons be if they all appeared independently? Now, common ancestry says two things share the same codon because they inherit it from a common ancestor. So that only had to happen once. So that's why the variation in the codon variation is much less, because individual species are inheriting codons from common ancestors. Whereas in the other model, each member of each family is appearing independently, and they have to be the same by chance, basically.
So that's what you're saying, that there's a...
30 seconds.
Oh, okay. Well, let me ask a question then.
Sure.
You know, talking about nodes, you did mention nodes and tips. Can you suggest a phylogenetic node that is actually, in other words, a missing link of any kind between any of the major groups of living things that would be not a fossil DNA, but an actual fossil?
Okay, that's 5 minutes right there.
I get 5 minutes, or is that up?
Well, I asked the question.
He was
I suppose you
asking
could
you.
refuse to
So now
answer it.
Or
No, I'll answer.
we
Yeah.
have 10 minutes remaining if you'd wish
Okay,
to just open
I'm just
back
getting,
and forth.
I'm just making sure I know what the deal is.
Well,
okay.
this has gotten too fun, and so, yeah.
So,
That's the,
Would you rather
nodes and missing links.
Would you just want to do back and forth for the next 10 minutes, and I'll let you know when there's 2 minutes, 1 minute.
Sure. You want to do that?
your turn. Pick a node.
So let's answer that question. Missing links is a terrible thing. It's sort of like survival of the fittest. It's just a very nonsensical kind of concept that gets spread around mostly by creationists in the public, and it doesn't carry the meaning that it has in evolutionary biology. So the way we think about life is that everything is a link to something else. Every individual is a link to the next generation. Populations are links to the next generation. Now the question is, do we look at the evidence and does it support common ancestry? Now let me give you an example. So I think I mentioned Ancestry.com, but how many of you guys have done Ancestry.com? Has anybody ever done that? Oh, you have? So, you know, when you do Ancestry.com is a good one because it'll go back and what will it tell you? It'll tell you like, you get like a pie chart from them and it'll tell you how much of your oh, you haven't done the DNA? Has anybody done the DNA? So you get like a chart and it tells you like what populations your DNA is from, right? So you're like, what was it?
It was like 43% Scandinavian, 37% British.
Yeah, that kind of thing. You're right. Now, did it tell you the names and addresses of all your ancestors going back several tens of thousands of years? No. So Ancestry.com is something that puts your genes in the context of a shared ancestry with other populations. So it nests you in a certain group. But that doesn't mean that it gives you a complete genealogy going back 50,000 years or whatever. It doesn't tell you the names and addresses of all these people. It just takes your DNA and puts your DNA in the context of these populations. So what we do is we look at how species are nested in other groups. Okay, so for instance, birds are a group that are nested within theropod dinosaurs. Not just any dinosaurs, but theropod dinosaurs. So they share a more recent common ancestry with theropods than they do with other vertebrates. It's like saying, when I went back to talking about you and your siblings and you and your cousins, you are more related to your siblings because you share a more recent common ancestor with your siblings than either one of you do with your cousins. You would know that if I had me, my sibling, and my cousins, and we didn't know our parents or grandparents, we would still know that pattern, even though we wouldn't know necessarily who our parents or our grandparents are. So you don't need that to test these models of common ancestry. So does that answer your question?
I asked you to name a node.
Nodes don't have names.
Don't they exist?
They are. It's a diagram that shows a relationship. It's the important thing about the nodes is it sets up the relationships between the things at the tips. So that's why siblings are more related to each other than siblings are to cousins, because they have a more recent common ancestor. That doesn't mean you have to put a name on what the node is.
Well, You mentioned...
What I like to say is the fossil record and genetics tells us evidence of common ancestry, but it doesn't give us genealogies. You don't look into the fossil record and find evidence of genealogies going back. You find evidence of common ancestry. Those are two different things.
Well, for instance, you mentioned theropod dinosaurs, and they gave rise to the birds, right?
Birds are nested within theropods. It's like saying we're mammals. It's like saying humans are mammals. So we had to evolve from a mammal.
Would there have been something, a unique organism that lived at the node stage of the story in the chart?
And it was a theropod.
Okay, it was a theropod.
Just like I can nest your ancestry in Northern Ireland, but I don't have to say who that great-great-great-great-great-great-grandfather is, that's your ancestor, but I can still put your genome in the context of Northern Ireland.
Well,
I can put birds in the context of theropods.
Don't forget that the design model would say that there isn't a node, that all that we really have is these, as you drew up here, and there wouldn't be something that, that both things came from the grandparent that made us cousins. So what I'm trying to say is, if there's nothing that's in the red part here, and all there is in the evidence or the data or fossils is the blue part, then I'm not saying these don't ever exist, but I'm looking for one that does. This would be for design, and the whole thing has to exist for descent to be true, right?
What you would get is if you compare those four groups, you would get consistent trees across. It's like all the whale stuff I showed that I breezed through. If you did that across different lines of evidence, you would get very similar trees across different lines of evidence every time. You wouldn't expect to get that if it was four independent lines.
Well, isn't there supposed to be something here at the node?
Yeah, but
like
you don't necessarily
a
know it's...
dinosaur bird or a
fish
that's turning into an amphibian
well,
or a simian that's turning into a human.
These creationist notions of there's like this halfway thing that's halfway this and halfway that.
Missing link. Yeah.
That's not what a missing link is. And that's why missing links are terrible
Then let's call it the
terms.
node. Can you give me an example of a node that has been found in the fossils, that there's evidence that it ever did live, that this red part was ever true?
We know theropods lived. And so the common ancestor of birds and theropod dinosaurs is some theropod dinosaur.
Well,
that doesn't mean we have the name and address of Mr. or Mrs. Theropod Dinosaur at that instant in time.
We have theropod dinosaur fossils and we have bird fossils, but what's the therabird fossil, the thing that's in the middle?
A bird is a theropod.
A bird is a dinosaur.
It's a specific kind. Yeah. It's just like saying that humans are mammals. We don't say humans are
There is
mammals.
no fossil you can actually name that is the change point. There is a turn in the road in the phylogenetic story, the history of these living things.
So because evolution is a branching, you guys ever see those diagrams that show like, you know, the guy's hunched over like this and gets a little taller and he gets a little, and then it's like some computer programmer walking at the end of it? That's a terrible example of evolution. That line like that, I hate that. But it's one that they show all the time, and I'm sure everybody here has seen it. Evolution doesn't work in a linear way like that. It's more of a branching process. So we can build these phylogenetic trees, which I explained already, that describe the diversity we see in some group, like this. So this branch here, so say this is all birds and theropod dinosaurs. This is an early branch of that group. So we would look at this group, and it would have a lot of characteristics that link it to, that are relatively old, say, or a lot of characteristics that link it to another group. For instance, a lot of the old groups of birds that branched off early had teeth, and they had a long bony tail. But they had other characteristics that were like modern birds. They had feathers that looked just like modern birds today. So it's those, we call those derived and ancestral character states. So instead of talking about species as missing links, we talk about that in terms of traits, right? So this mosaic of traits. So these early branches would be indications of what the ancestor might have looked like. But we don't have, you can't never know what's really at that node, but you can build this pattern that explains the data. So saying what's at the node is sort of like saying that in your Ancestry.com report, you're demanding Ancestry.com to tell you exactly your great-great-great-great-great-great-grandfather in Northern Ireland's name and address. It's a bit like asking that of the evidence.
Do I have any time left?
We have a minute and a half left in this.
Go pull up DinoBird node. There's not been the naming of some kind of a name of an organism that actually is the common ancestor. If we're talking about common ancestry, if we're talking about common descent, there must be a common ancestor. So yeah, DinoBird node. It's the blue, it says in big white letters, Dino. There are many different DinoBird suggested fossils that could be at the nodal points.
1 minute.
Could have gradually, okay, next. Okay, and this was from 1998 National Geographic, and they lined up all these different fossils, and it looks like a progression going through common ancestry. But when you look in the article, this one's actually dated by descent methods 120 million years old. This one's 90 million years old. That one's 80 million years old. Now we're 120 million years old again, 120 million years old. And this one, the Archaeopteryx, the bony-tailed one you were talking about, is dated at 150 million years old. Where did I get these dates? They were in the exact same article. This is when a whole bunch of these fossils had recently been found over a number of years. In 1998, National Geographic had a special issue on it. The dates are from the same article. I didn't make these up. This is what it said. They're actually in the wrong order. They look like a story of something that's happened, of common descent actually progressing, and they're in the wrong order because it looks better that way. And looking for some fossil species, is what really is the
And that
hard
explains
rock
why
fossil.
that chimpanzee to human line thing is so terrible. And he just explained it right there because it's a bush and not a line.
All right, now we have our 5-minute closing statements. Appreciate you, gentlemen. See, we can have these conversations, okay? So, and who knew that you had to take statistics to learn all this stuff?
You have no idea.
Oh, I think I do. That's the scary thing.
It's bad.
Yeah. You notice that a lot of the discussion was over statistics tonight. So when you deal with large populations, you'll wind up getting statistics in the mix. So let's see. Dr. Mays went first. Would you like to go first, sir?
Oh, sure. That 5-minute thing.
Yeah, Dr. Jackson, your 5 minutes. All right, thank you.
Is that okay, Dr. Mays?
Oh, yeah.
Oh, okay.
Yeah, sure.
Well, yeah, I just want to round off something. There's new data, new information, new facts, not alternative facts, that have come in. And I'm not using this in my step-out presentation because it really needs to, it's brand new, it needs to be confirmed by more people. And yes, it's statistical. New article coming out, PhysOrg. Again, this is not a creationist, it's an evolutionist, peer-reviewed by common descent peers. And they came out with this in just May of this year. Sweeping gene survey reveals new facets of evolution. This researcher from Rockefeller University and another one, David Thaler from University of Basel in Switzerland, published this. And it says it's sure to jostle, if not overturn, more than one settled idea about how evolution unfolds. It is textbook biology, for example, that species with large, far-flung populations, think ants, rats, humans, will become more genetically diverse over time. But is that true? The answer is no, says Stokel, lead author of the study published in the Journal of Human Evolution. For the planet's 7.6 billion people, 500 million house sparrows, or 100,000 sandpipers, genetic diversity is about the same, he told AFP. The study's most startling result, perhaps, is that 9 out of 10 species on Earth today, including humans, came into being 100,000 to 200,000 years ago. And that would go with design, of course, it depends on when the design occurred, but that all of it occurred at the same time, or 90% of it did, sure leans towards design instead of descent, taking so long to get these things. Now, some explanations. were that maybe some horrible cataclysm sort of had all living things go into a genetic foray and have to reinvent or reestablish the genomes. But that's not something that we know yet. But when I'm sorry. Could you click it one more, please? Yeah, okay. What they saw was a lack of variation in so-called neutral mutations. Since there wasn't very much variation, they were assuming then there wasn't very much time, because it would take time to make those variations by the transposons and things like that, and mutations. But they saw a lack of variation in neutral mutations, which are the slight changes in DNA across generations that neither help nor hurt an individual's chances of survival. And yet, another unexpected finding from the study, species have very clear genetic boundaries, and there's nothing much in between. Now, there's not a mathematical maze of statistical workups for this. They're actually just doing the same kind of DNA fingerprinting things that they do, and they adopted the same methods that are used in making sure that smoked salmon and sushi and things like that are actually the genuine article. You can read more about it here. There's the link. Gene study reveals facets of evolution. And it says there are clear genetic boundaries, and there's nothing much in between all of the species. If individuals are stars, then species are galaxies, said Thaler. They are compact clusters in the vastness of empty sequence space. The absence of in-between species is something that also perplexed Darwin. And those in-betweens are links between, and the term missing link became very popular, transitional form, node, whatever you want to call it. There has to be some stepping stone in the common descent story. There's got to be a grandpa or the cousins to be really commonly descended from a common ancestor. And so this is new. I'm not going to use it as proof, because I need to see a verification. We need to see this study replicated, but it's respectable, and so maybe in the next time, either myself or Dr. Mays are speaking to a group, we'll have a common design and or a common descent take on this new information.
Oh, yeah.
Oh, sorry.
That's okay. So first, I really want to thank everybody for having me. It's been great. I think everyone should talk about stuff, and especially about science. Everybody should talk about it, and especially, I think my job as a scientist, especially someone that lives at least in your region or your community, is to be accessible to you to answer questions. So let's start with what common ancestry is not. So it's not a model that has anything to do with the origin of life. That's a separate issue. It's not a linear progression, just like we talked about. It's not sort of less complex to more complex. It's not individual organisms turning into other species. It happens sometimes, and you can ask me about it later, but that's not generally what it is. It's not a different species mating to make new species. It's not necessarily to have complete genealogies to support common ancestry either. And it's not like an argument of like begets like doesn't contradict common ancestry. These are things that creationists say it is, but it's not. No scientist says that. So can a designer explain the data? Yeah. An omnipotent designer, God, can do anything. And an explanation that can do anything, I can't test. That doesn't mean it's not true. That doesn't mean you can't believe it. I just can't test it. Because if your explanation can take into account any conceivable data, I can't rule it out. So designers of course they can explain everything I've said, but if your designer is an omnipotent God, then it's not science. I can't test it. That doesn't mean you can't believe it. I'll just skip it. So what's the harm here? So what's the harm to science? What I've been expressing to you is this is what scientists say. Nothing really that creationists say is really in line with what actual science is saying these days. It's just not true. So what's the harm to science? Science denial places our technological and scientific progress in jeopardy. This isn't happening in China or India right now, and I've been to China, I know that. Science denial challenges the growth of students in their education and their careers. And science literacy, especially in the public, is important. So even if you don't believe it, you should know the difference between what scientists say and what they don't say. So what's the harm to public policy? Well, you need facts, and you need to know what the science says to make important decisions. And erosion of trust in our public institution erodes our health, our prosperity, and our safety. Now, this is a question for you guys. What's the harm to your faith? Most religious traditions, there's no doctrinal commitment to denying evolution. Religious fundamentalism reduces faith to sort of a materialist philosophy that I'm only going to believe what's literally true. And here, science denial positions distracts from good missions of charity and good works. Science denial also turns people away from faith, especially young people. So here's a survey from the Barna Group, and this is in a book by David Kinnaman. These are reasons that young people are turned away from the church. And 35% say Christians are too confident they know all the answers. And they say, about 29% say churches are out of step with the scientific world. 25% say Christianity is anti-science. That shouldn't be the case. That simply shouldn't be the case. So this tile that I showed you that's on the floor of Notre Dame, this saying was written by an Eastern Orthodox Christian. Head of the National Institutes of Health right now is Francis Collins. He accepts everything that I've told you today, and he's an evangelical Christian. These things are not incompatible with one another. It's okay, even if you're just starting to think about it, it's okay to start thinking about it. It's not going to threaten your identity as a Christian. So are you convinced? I don't think so. So if you view the acceptance of common ancestry as a threat to your group identity, so if you feel like, I can't be a Christian if I believe this, then nothing I say in terms of the evidence is going to convince you. Nothing. So what I suggest is you look at places where like-minded people, people in your community, people that share your values, that share your faith, who are also accepting of the science of evolution, and talk to them about how it works. Because it is an important thing. Thanks.
All right, folks, if you don't mind, let's have another round of applause for Dr. Mays and Dr. Jackson. Now we are very quickly going to set up for a question and answer period, and we will have two microphones. We'll have two lines. One line is for Dr. Jackson. The other line is questions for Dr. Mays. Oh, one right in the center? Right here, okay. That's fine. I'm about to get rid of the mic here for those who wish to.
Or they can just ask from where they're sitting and pass the mic. Oh, okay. However you want to do it.
Do we have folks interested in asking questions? I see some hands going up, okay. Are you about ready, Chan, for them to make their way up here? Okay. I'll tell you what, we could have them queue up in two lines and I can hand the mic if you wish. We should do it that way.
We're the ones that have to be on TV.
Yes.
The internet TV.
All right. Tell you what, if yeah.
Make him come to you. You don't
I
have to
would
get
say
up.
make Chan come to you.
Yeah.
All right. All right. Is this working?
All right. This is for Mr. Jackson. I understand. I mean, I hope you didn't see me. I wasn't trying to be rude, but I was trying to look you up a little bit. I'll speak up. I was trying to look you up a little bit to make sure that you were Christian and creationist. And it even said somewhere you went to Croatia for like some missionary work. Okay, yeah.
Well, I spoke at the University of Split there 4 times in Croatia, yeah.
Okay. But my question is, I don't go nearly as deep as you guys do into this. Like that's not my job. So I kind of trust the scientific community to do their best with what they have. But if tomorrow, let's just say, they turn around and go, oh no, this supports, common design. And I go, okay, fine. But then all of a sudden, I don't know if you would if you would propose this, or because I certainly know others would, of saying, okay, now Christian God is real. I would have to go, this doesn't support, how do you know it's that God versus another deity or versus a God we may not know about? I guess I'm trying to see the parallel between one particular religion versus in this idea of common design, how, how I guess disproving evolution will, in a way, and fighting support for this will lead to one
Who's
religion.
the designer?
That's what I'm trying to say.
Okay.
How do you know that?
Yeah.
Is this a question for me?
Yes.
And what I heard was trusting a scientific community, which I am a part of the scientific community. I reject the idea that because I am not a common descent person, that I'm not a part of the scientific community or that I'm somehow anti-science. Yes, I'm anti the model of common descent, but I'm not anti-science. I've been teaching science since 1980.
Well, I hope I didn't imply that. I was just saying what the major, 98% was.
I wasn't thinking that you were implying that, but I wanted you to understand that that there are many of us who, have doctoral degrees and all of that and are into fields of science or teaching science who are in the scientific community as far as trusting the scientific community. Now, the majority rule or the consensus might never swing to common design. If they did, that doesn't mean that you're right. It doesn't mean that there's any particular religion. Don't forget, and I think Dr. Mays mentioned it, the only thing you got to do to be a card-carrying creationist is believe there's a creator. I mean, that could be Thor, or it could be Jehovah or Allah or whoever. But the implication of a scientific position, yes, it does have theological implications. The scientific community actually can't dictate your religious beliefs, but we can bring, and that's what we've been both doing, bringing concepts, bringing interpretations, and also bringing data and evidence. Like I said, not proof, but evidence. And so, yeah, I can see you're watching it happen. You're not as into it deeply. And trusting the scientific community, well, it's like trusting politics. Is someone good just because they got all the votes? Is something true because the consensus says it is?
Don't lump me in with the politicians. That's an insult.
Hey, I was talking about science not really being like politics. So, yeah, I know politicians, that's a whole nother thing that I'm sure you and I both have disdain for. So I see what you're a spectator sport onto this thing. I'm not sure exactly what your question was. I hope I kind of reacted to it. But if you had a real specific question like, does common design mean Jesus is real? I'm not sure what you're asking.
No, you actually did answer my question very well. Thank you. That was kind of satisfying to hear.
I also have a question towards Jackson. I remember several years ago, I think I was back in high school, I watched this documentary about how they took this chicken embryo and just by simply tweaking its genes, it was able to give them teeth and a tail. And I looked into that and they found the reason those genes are in there is because they share a common ancestor down the line with reptiles and crocodiles. And the same, you can see similar things in humans. Like, for example, the existence of goosebumps is because at one point we had hair, and the goosebumps allows our fur to, sprout up and make us look bigger for danger. So with that in mind, how does your model account for the existence of those genes?
Well, as Dr. Mays mentioned, some extinct birds did have teeth. That does not necessarily mean that they are reptiles. Things that have lost their teeth, there are mammals that don't have teeth. That doesn't necessarily mean that they're not mammals, or that there's a common ancestor for that, and it proves that they came from reptiles, for instance. As far as and what was the second thing you mentioned?
Goosebumps.
Oh, the goosebumps, yeah. The function of the goosebumps, or think of it, when do you get goosebumps? When? What happens? There's an emotional thing.
When I speak in public, like right now.
Oh, okay. Like, if you're nervous or scared, or maybe if a cold breeze blows on you, then you get the goosebumps. For animals, they get that when they're angry. They fluff up and they look bigger and growl at you. It's two different. Now, both of them may have something to do, well, certainly with posturing for like a coyote or something like that. For humans, it's a different emotion that engenders this. We do think that it has something to do with temperature regulation in the skin, but saying that means that dogs and people have a common ancestor because of that, there are other explanations for it. And for you to be able to say, well, my explanation is the one that's true, you have to find some condition, some situation, some data that could only be true if no other thing but your theory predicts it. That's how science works. If something else could say, like I could say, well, common design is true because it predicts the sky would be blue. But there are many hundreds of other theories where the sky being blue would be part of it too. It has to be something that's a unique prediction in how science works. So I hope I answered both those questions.
Yeah, pretty much.
My question's for Dr. May. Since you're in academia, a common criticism that the creation side would levy against academia is that you never have consensus, or you can't have a fair consensus because those that would accept a creation model that we would think of, particularly in the Judeo-Christian community, is shut out or excluded from that. And I guess my question to you would be, if you were sitting on a tenure committee or a tenure review board for a colleague, and that particular colleague had in their portfolio publications or materials that tended to support a young earth creation type view, what would your position be, and how would you react to that as a member of that tenure review?
So first of all, I mean, obviously people in science aren't shut out of science because they're Christians, because I have colleagues who are of all kinds of faiths that don't get shut out of academia just because they're Christian. Like I said, Francis Collins, Theodosius Dobzhansky, evolution is part of the religious schools, like Notre Dame is a Catholic university, so Christians aren't shut out of academia for being Christians. Now, if someone says any position, so I'm not biased against creationism per se, I'm biased against any position that's not supported by the data. So the data determine my biases. So if someone is attempting to push an idea that has no support, then that's what I would be against. But that has nothing to do with whether they're a Christian or not.
In the data would be peer-reviewed articles and publications and things that are accepted.
Well, my job as a scientist is to convince my peers of things, and that's a tough job because we all want to one-up each other. It's not nice. It's not like I don't get papers accepted into journals for my research just because I believe in evolution. It doesn't work that way. It's brutal, right? So everybody's being critiqued, and you have to compete on that field. There's no set of rules that are going to change for you just because you don't like the rules. That doesn't mean you can't be a Christian. And you can be a Christian and be a scientist, and many, many, many are.
But one of the rules would be walking your paper to get published across that tile that you showed in your slide that says basically the foundation is evolution.
Sure. And you would also be walking past, I could imagine you could put other tiles in that building about how matter is made of atoms, and that the sun is at the center of the solar system, and germs cause disease. All those are well-established ideas. If you want to overturn them, that's fine. But ideas that become that well-established over time are it's a tough nut to crack to overturn them. So it's not just that.
I had a question for Dr. Jackson concerning your presentation. Can you hear me okay? Okay. So during your presentation, you gave 3 criteria that would act as predictions for both the, let's say, common design model and common ancestry model. And for the common ancestry model, you said that there would be a prediction that you would make, or that you would have to make, is that there would be junk DNA. And I think it's pretty clear that Dr. Mays addressed that by giving an example of an amoeba having 100 times size the genome of a human being, which you never did address, by the way. You never responded to that in any way. But more importantly, the question I wanted to ask is, in your own model, you said there would be no junk DNA, there would be no junk structures within this kind of common design model. And yet, you, I think, within the next sentence, and you said this a few times in your presentation, said that there are some corruption of the structure and there are some corruption of the DNA by things such as mutation. Obviously, any reproductive process is going to create mutation in the genes. And so, in a sense, have you not disproved your own model if you're saying no junk DNA, no junk structures, but then in the next sentence saying, well, but mutation does cause some corruption of the structures and it does cause this less than perfect structures, right? Have you not disproven your own model and that this prediction has not been satisfactorily, cannot be used to support your own model of common design?
Oh, no, that's quickly answered. I wasn't contradicting myself at all. It was completely conducive with design, is that everything was set up very, very highly designed at first, and that there could be corruption along the way, but not that 97% of the genome would be corrupted. In that sense, you'd think that, well, death would have occurred, extinction would have occurred in that germline a long, long time ago. I'm just talking about we would expect everything would have been functional at the beginning. And yes, copying errors, mutations, and other kind of damages that would have occurred along the way. Some genes could have become non-functional, like we talk about the pseudogene of humans not being able to synthesize vitamin C. Fruit bats, primates don't synthesize vitamin C, but all the others do. We're talking about this vitamin C pseudogene. Maybe it's an old gene we have that used to make vitamin C and it doesn't work anymore. But as long as we eat fruit, we'll be okay. But maybe the designer designed primates to eat fruit, and fruit bats too, and so we didn't need to have a gene, and it just kind of looks like the vitamin C synthesis gene. So what I'm saying is that the preponderance of the genome would have, with 100% would have been functional at the beginning, and any pot shots that had been taken at it over the decades, over the millennia, by mutations could have caused some damages, but not on the whole scale thing that we would expect non-functional because they don't need to function and never did have a function after we descended into something new. What I'm trying to say is the design model would only predict a little bit of this, which is what we find, not a whole lot of this non-functional junk DNA and junk organs like the descent model.
So your contention is that it's the quantity of the corruption that
Sure.
you have a problem with. Because it's what's interesting to me is that I don't think Dr. Mays would argue that, say, the original common ancestor, for example, had corrupt DNA. I mean, the original, say, you know.
Well, I wouldn't call any of it corrupt. It just varies.
Right. I mean, so
we describe the effect of that variation on the organism.
You're saying that the original ancestor that was created by a designer has, you know, this perfect genome in a sense.
Yeah.
And I don't see how that would be different in any way from the common ancestry. It would be perfect in the sense that it was the first one, and that all genomes, you know, were delineated from that point. Do you see what I'm saying?
No, because what you said, I said, was that quantity mattered, and that's not what I meant at all. It's not quantity; it's quality. In other words, it's where did all this non-functioning stuff come from? Did it come from damage, or did it come from a history that's now lame, that isn't used? What the nature of this? What actually caused these genes that weren't doing anything anymore? Was it because they used to and they're damaged, which is what the design model would say, or was it because these have now gone on gene duplication and then tinkering by mutation and natural selection to arising new genomes, and this is the leftovers? You see, I'm not saying that it's sheerly quantity. I'm saying it's what was the origin of these massive parts of the genome. The descent model would say there'd be a lot of this. The design model would say there'd be only little of it, and it wasn't that it stopped being used, it's that it stopped functioning.
All right, thank you very much.
So my question is for you, Dr. Mays. I'm about to go to college in a few years, and let's say go to college X, and they are not Christian. Let's say after I'm in biology class, and the professor asks me to write a paper about creation and why it is false. But I'm a Christian, and I think that everything about that is wrong. How do I write that paper and still get a good grade, because my grade could determine what I do, but still get a good grade on it?
Well, no professor should ask you to write that paper. I wouldn't ask you to write that paper. Why would I ask you to write that paper?
To see my viewpoints and to see my, what I could do.
Your professor, but in a biology class, a biology professor is going to be teaching you biology, right? So he's just going to be telling you, this is the science of biology. You're there to learn it, and you can believe it or not. That's fine. In fact, your professor might even be a Christian. In fact, all likelihood he probably is, he or she probably is. So I can't imagine why they would ask you to write that paper.
Well, in biology, it could teach you about carbon-14, and it could say that carbon-14 was billions of years old, and I would have to write a paper about said carbon. But I'm like, well, that's not true. But I would still have to write a paper or do something with that. But I would say no, and then I would get a bad grade.
If you say no to anything that you're asked in science to do, because what I would ask you to do is support it. So I would say, okay, you don't think carbon-14 isn't really billions of years. All atoms are billions of years old, right? So if you don't buy that, though, then the paper you should write should be one with evidence to support why that's not the case. And if you have the evidence and you can convince people, you're golden. If you don't, then you haven't done a good job convincing people. That's it. And again, I wouldn't ask you to write that paper. I wouldn't ask you to say, write a paper about, why creationism is bad or something. I wouldn't ask you to write that.
Okay, thank you for your time.
Yep.
All right, so this is a question for you, Mr. Mace. You see the common ancestry thing, and you used Ancestry.com. Mm-hmm. To me, that's a family tree. It's tracing your family tree based off of what info you give. So if you take your common ancestry and put it next to a family tree, it's almost as complex as a family tree. Would it not be?
Your common ancestry and put it next to a family tree?
Yeah, it would be.
Well, your family trees,
the same complexity, right?
When you get there's two things from Ancestry.com. One is a genealogy that you get, and they do that based on historical records. That's going to be very limited, and it's only going to go back so far, right? So that's going to be a very tiny little slice of your ancestry. It's not even going to be all of it. Because think about it. You have 2 parents, right? They each have 2 parents. So you have 4 grandparents.
Yes.
How many? You have 8 great-grandparents. You should have 16. So it gets bigger and bigger. Now, it doesn't always get bigger and bigger because at some point there's a little, what we call inbreeding going on. So it doesn't just keep going like that, right?
Correct.
So what you get when you get the genealogy you get back, the history, is only part, if you trace back a line of all those ancestors, it's only a few little lines. It's only a little bit of that story. It only goes back so far. The other thing that Ancestry.com does is you can take a little swab of your cheek and you get DNA and you send them that. That, when they look at that, is exactly the same technique. What they're doing is exactly the same technique that we would use to look at, say, the phylogenetics of whales and deer and other mammals and things like that. It's exactly the same. And so they would take your DNA and put it in the context of this historical pattern that goes back much farther than what you can trace a historical ancestry on. So the two are very different things. One is much deeper, and it places your genome in the context of existing genetic variation in human populations. One is very specific little lineages that only go back a certain amount of time.
All right, so, and if we take the genocide part of it, you take any point of anybody in your family, say your great-grandpa, you use that as a starting point, and you branch off of that. Wouldn't that be the same as doing that for a species? Of putting a species, you said you couldn't name a node, but to me, I think you can if you're using that.
Because your great-grandpa, you know who he is. You can go to him. Most of us know our great-grandpa, have met him, have met our great-grandpas. I've met my great-grandpa. You met them, they're people. They have a historical record. There's another source of information for that outside of DNA that allows you to establish who that ancestor is. The other one, there's no other source of information. 50,000 years ago, they didn't have the county courthouse where you could go and look up records, right? So in that sense, you don't have the same information that you have for an ancestor 50,000 years ago as one 3 generations ago. So they're really 2 separate things. You're only going on, you're using a different method to get common ancestry. And also, your genome it undergoes what we call recombination. So some of your genes come from some places, and some of your genes come from somewhere else. And it's all mixed up. So a lot of people, Americans have a very mixed genome. They have a lot, some of us have a lot of genes from Northern Europe. Some of us have genes from Africa that are pretty recent in our history. Some have genes from Asia. Some of us have genes from Neanderthals that go back really far. And in fact, at Ancestry.com, you can see what proportion of your genome came from Neanderthals. And in most people in Eurasia, it can be anywhere from 1 to 4%. So, but Neanderthals don't have county courthouses, so we can't go back and find their records to say that Neanderthal was your great-great-great-great-great. Can't do it.
Correct, but a known would be a starting point for, say, from a theropod to a bird. That known would be a point of conversion, correct?
That node is a hypothetical population that existed in the past that links 2 lineages together.
All right, so wouldn't that be, in a sense, the same as your relationship to your grandparent?
Well, sort of, except that because the branches that we're looking at are not individuals, like you and your cousin, at the tips, they're whole species, so they're whole populations. So the node is a whole population of organisms there. It's not just sort of one like it would be in a pedigree, in your genealogy. They're a little different.
But a node is, you're putting a species as a node, correct?
Yeah, there would be a species there, but it doesn't mean you know what it was exactly. But still, what you have is you have a test of the common ancestry model, so you know 2 things share a common ancestry, but you don't have a genealogy for them. If I don't have any information about your parents, let's say you don't know, you and your siblings don't know your parents, you never met them, you never met your grandparents, you never met your great-grandparents, I could still group you and your siblings and your cousins and your second cousins into a graph like that. I don't have to know exactly where they came from. I can use your genetic information to group you together like that without knowing exactly who they were, even though they were there.
Anybody else with questions? All right.
Okay, this is for Dr. May. I may show my ignorance here. But,
That's no, okay.
You mentioned the bit about removing a chunk of DNA from a mouse and not killing it or something like that. Like, how would you remove just a chunk of DNA, and like what are you describing there, like in a lab process?
I would have to go back in that paper and tell you the details, but I can tell you in general how that happens. Organisms have mutations. So there are mutations that happen all the time, and some of those mutations involve the deletion of big blocks of your genome. We have these mutations in us right now. Not all of us here don't have the same amount of DNA. Some of us might have a big chunk of DNA missing. And it makes no difference. So what they did is once they found these mice that had this chunk deleted, they breed them, and they create these lines. So now that they breed them all with each other individuals that have the same deletion, the same mutation, they create a line of mice that have this deletion in them and used for research. So one thing that's convenient about things like fruit flies and mice is they breed like crazy, and they breed really quick, and they're cheap to keep in a small space. So you can make lots of them, and then when you find one with an interesting mutation, you can say, we want to keep that, so let's breed them with their siblings and stuff. I know it sounds, but the mice, they don't know the difference. And well, they do sort of, but it's another story. So then you can create this line of mice that has this deletion now, and now you can go and use it in research.
Okay.
So it can be just a mutation that pops up.
Okay.
Or you can go in and snip, or you can go in now people can go in the embryos of the mice and manipulate their genome and take out things or change the sequence and see what happens. So they can do that too.
I'd actually like to address both of you partially on this question. With your model of descendants and nodes and such, it all kind of stems from one single root. What do you have an identity or a specific animal or whatever that supposedly roots all of that? And kind of a follow-up to that, if we all descend from one single point, what is the purpose or what is the explanation of the amoeba having 100 times the amount of genes or DNA that we do?
So that's what we call the LUCA. LUCA stands for Last Universal Common Ancestor. So we can test ideas about what the LUCA looked like by comparing existing organisms today. This last universal common ancestor had to be microbial. If you go back far enough in time, there was nothing in the fossil. You don't go back in time to 3 billion years ago and find like rabbit fossils and things like that. So it had to be a microbial organism. So it was probably a community of microbial organisms, however. It wasn't just one. And that's because microbes do something really interesting. They can swap genes with one another. So you can imagine this big community of bacteria. They're not like pairing up husbands and wives and having sex and having kids like we do. Instead, they are just throwing their DNA out there everywhere, and microbes are like, I'll take that, I'll take that, I'll take that. So if this big community of organisms is swapping genes, it creates a lot of variation. And so that's probably what we think the first organism looks like. And then, the amoeba question is, sorry to interrupt you, but the amoeba...
Can I, before you address
that,
can I have a follow-up? But if there was just one organism, how was it swapping with many others if there was a community?
Because bacteria, we know this of bacteria today. Different bacteria of not just different species, and species is a much easier thing to define in sexually reproducing organisms, much tougher to define in microbes. Whole different families, classes, whatever, of bacteria will swap genes with one another. So it's as if you walk outside and you pick up a gene from an oak tree and now you can photosynthesize. That's the kind of things that microbes do sometimes.
Yeah, but if
So there were lots of different species interacting, and they're evolving over time and changing.
But didn't they all have one root? Is that kind of what you were?
They might have different lineages, right?
But did they all root from one thing?
So the root is a lot more like this. So instead of one root, what it is you have a lot of roots, and they're reticulated, where
And
the
now
genes
you're starting
are.
to look like his model.
Yeah, except they're all connected back here, but they're swapping genes. So you can go back to one root, but at the beginning it's all like this, and then they start going off into separate lineages.
So there wasn't one root thing at the bottom.
Yeah. So you can test the papers I showed up there that tested like common ancestry. They use statistical techniques and molecular biology to test common ancestry models in like primates. People have done similar papers that look at bacteria, archaea, and eukaryotes. Those are everything on Earth that's alive is one of those three things. So they did this to test for ideas about universal common ancestry, and they found that the data support the idea that those three things, bacteria, archaea, and eukaryotes, we're eukaryotes, by the way, are all connected to one another in a common ancestry.
Is there an explanation for where those came from?
Well, again, like I said before, the origin of life is a tough question. Okay. And so the origin of life is something that people are working on. It's not connected. So if you want to say, let's say for argument's sake, God made the first life. That doesn't stop any of these other models of common ancestry from working when tested against the data. Now, that's a fine belief, but it's not a scientific hypothesis, and people are testing hypotheses about the origin of life, but it's an open question. So the amoeba, those big genomes are a lot of times because of gene duplication. So a lot of times what you get is in a lot of these organisms, they have big segments of their DNA that gets replicated over and over again, so you have all these extra copies of it laying around. And that has a lot to do with a lot of different things. But that's basically what it is. They don't necessarily need all that other DNA. You don't need to have 100 times the DNA, than a human to be an amoeba. That's for sure. But that's usually how it arises through mutational events of duplication. Did that help?
Yeah, and your answer to why the amoeba have 100 times the DNA that we do is, could you restate that?
Well, just like what Dr. May said, ferns have 400 chromosomes. That doesn't mean that they're more advanced than us.
I didn't say amoebas are more advanced than us.
Oh, no, I was answering his question.
But they're not, that has nothing to do with advanced or not advanced.
Well, I think nobody in the room thinks amoebas are more advanced than us, and I certainly don't.
I just wondered, since you said advanced.
I don't mean to be prejudiced against amoebas. How politically correct do I have to be? But I mean, having a lot of genetic material in your nucleus doesn't necessarily mean you're rich in genetic information. And if it's just from a bunch of gene duplications, which are sort of co-opted into descent theory about how new genes can be formed. A duplicate is made, and then natural selection and mutation work on one. That's the theory of how that story would go. But just having a whole bunch of copies of the same, like a jellyfish DNA, 4 copies of that, doesn't make it more information. It just happened because these organisms, again, like Dr. May said, for the microorganisms, their DNA is a whole lot more loosey-goosey than ours. I mean, they pump out copies of their DNA in little spaghetti-o shapes called plasmids, and every bacteria on the planet can share it with each other like Pokemon cards, and they're all between species, and so they generate their own little community of genetic information, but they could get a lot of information they don't need. The amoeba just has a whole bunch of copies of the same thing. It doesn't use all the copies. It just only needs one copy of something it has way too much of. So this is kind of just a quirk of the genetics of an amoeba, and really is kind of transparent to the issue of whether whether design theory or descent theory is true.
It's bacteria that have the plasmids that swapping around, not amoeba. Amoeba are eukaryotes. They're not
they're
bacteria.
eukaryotes, but
they're more like us than they are bacteria. Yep.
All right, am I good?
Good.
This is directed to both participants. I understand what you said about I forget the specific dinosaur, transitioning to birds being fairly difficult to find the node or common ancestor, whatever you want to call it. But my question would be more towards parallel evolution. How much easier, if it is, would it be to find the common node or the common ancestor? Here's an example. Like, I know this may not be exact, but let's take today's monkeys, the bonobos, the chimps, and the humans. Let's say that's parallel evolution. What would make that harder than to find the common ancestor for that, or would there be any difference at all?
So there's two things. There's two ideas in evolution. There's parallel evolution and there's convergent evolution. So convergent evolution is like if you see like a dolphin and a shark both have like a kind of roughly triangular shaped dorsal fin, right? Well, they developed that independently of one another. So that evolved so that they can stabilize themselves because they both live in the same environment. But it's not the same structure. It's really different in how it's put together. Parallel evolution is when two organisms develop kind of the same trait independently of one another. It's not just superficially the same, it's really the same. So what you get, in fact, if I'll show you later on that list of books that I had, there's a really good book there called Improbable Destinies by Jonathan Losos, and he's studied lizards in the Caribbean on different islands. One of the things they find is you do get a lot of parallel evolution, but it tends to happen in species that are very closely related to one another. So they already have very similar genes. So if you put them in the same environment independently in two different islands, and they're closely related, they tend to evolve the same adaptations in those environments. So sometimes convergence in general can be a confusing thing when you look at phylogeny and you want to determine relationships. That's why we look at these traits that are neutral, because traits that are neutral, that aren't influenced by selection, are going to be less prone to convergence. And so we are aware, scientists who do this work are aware of different kinds of traits that we use to study relationships, and we can parse out, okay, this trait is neutral, so it's going to be a better marker of history of a relationship pattern than another one that we know is subject to strong convergence. So we can parse out those different kinds of traits when we're doing phylogenetics.
Well, and as far as common ancestor and parallel evolution, well, the design model doesn't have that. Yeah. But the octopus eye and the human eye being so similar would be a feature of the design of each of them, not that they were related to each other or that their eye comes from a common ancestor eye.
Except they're not. So the eye of an octopus and a vertebrate eye are very different from one another.
Well, but
it's more convergence than it is parallelism because they developed similar structures, but if you look at the structure of the eye internally, it's actually totally different than a human eye. So they're not really the same.
They're not really similar?
They're not that similar, no.
Oh, Google it. Go ahead.
I don't have to.
I didn't mean you.
Any more questions? Yeah, one more.
I'm timing you at 2 minutes, though.
Yeah, cut him off if he goes too long.
Not going to sabotage the debate. One thing, you had talked before when we were talking about junk DNA and the kind of expectations that you would have, say, on the design model and the common ancestry model. And you had said, in my last question, you had responded, well, we would expect on a design model to have, this amount of corruption over time. But my question to you is, if that's a scientific hypothesis, you're saying this is a scientific hypothesis that in the design model we expect this much corruption over time, would you not need to know the intention of the designer to know how much corruption to expect over time? I mean, it makes perfect sense that we would know how much corruption to expect over time, for example, or how much mutation to expect over time in the common ancestry model. But if you are saying that you know how much corruption to expect over time in the design model, does that not require knowing the intention of the designer to have a reasonable expectation? And if you don't know the intention of the designer, then how can you use that as a scientific hypothesis?
Well, no, you're kind of switching this all around. The intention of the designer was for the genome to work. Okay, that's obvious. But we're not saying that the designer intended some certain amount of corruption. What makes the certain amount of corruption is just how bad it can get before a genome becomes non-functional and a genome line becomes extinct. There are some people that think maybe the dinosaurs became extinct because their DNA was just more susceptible to the kinds of corruption that we're talking about. So no, the degree of corruption is just how fast mutation rates occur, accidents, mistakes, copying errors, and then how far that can go before reproductive function no longer works or survival no longer works and extinction happens. So it's not like we're quantifying, we expect this much genetic meltdown, this much genetic dysfunction. We're just saying we would expect the majority of the genome, all of it would have been functional at first, and that everything would just be degrading over time. How far? Well, I don't know exactly how far. I know if it goes too bad, extinction will occur in that lineage that started out from the beginning, and now, for whatever mysterious reasons, maybe the dinosaurs, for instance. So no, I wasn't saying I know how much corruption or that the model predicts how much, just that there will be some, but that it won't be huge whole scale, like 97% of the DNA is just not going to function anymore.
But do you deny that you are assuming the designer's intention in the fact that there is corruption? I mean, clearly if the designer designed corruption into the DNA, into the process of reproduction.
Ford doesn't make cars with the intention that they're going to rust, any more than a designer would design a genome with the intention that it's going to corrupt. It's wear and tear. You see what I mean by that? There's not a single strand of any notion that the designer had programmed in some kind of corruption. It just happened, like rusting of a car.
Okay, thank you very much.
Yeah.
All right, well, I would like to thank all of you for coming out this evening. You have a special safe trip home, and I would like to thank the debate planners and Dr. Mays and Dr. Jackson. If you noticed, the topic was very narrow tonight, very, very narrow, and you think, seeing how this is a church, the things discussed in church typically get very broad when you're discussing creation, Genesis chapters 1 through 3. Think of the topics that weren't discussed tonight, and think how narrow this discussion was. And we just stayed on the issues of the life we see on this planet and how we got where we are. And so I would ask all of you to have a safe trip home. Thank you very much. And one last round of applause for our speakers.
And just remember, I'm just down the road at Marshall in Huntington, and if you ever have other questions, I feel like I'm there as a community resource for you. So feel free to email me or stop by and ask me.
For all those who appreciate the work that we're doing here on Standing for Truth, please hit that subscribe button because we are just getting started.